Hi student, I am Dr. Thaware. Now I share the screen of today's lecture in front of you. Please wait. Okay, welcome to the 11th lecture of course number Path 354. Title of this course, that is diseases of field and horticultural crops and their management. Okay, so in today's lecture we discuss about virus disease seen in okra, that is yellow vein mosaic disease of okra. Okay, so this disease is the most devastating disease seen in okra. Okay, so we see the some historical concept regarding this disease. So Kulkarni scientists first time observed and seen the viral infection seen on okra crop, and finally confirmed the virus produces the disease in okra, that is yellow vein mosaic disease. Okay, so this Kulkarni scientist reported this virus on bhendi crop or okra crop in Bombay in 1924. Okay, so this disease is most common found in all the subtropical region in the rainy season as well as summer seasons. Means this infection generally seen in kharif crops as well as summer crops. Okay, so due to the virus infection seen on the okra, so yield losses is up to 50 to 80% generally reported in all the susceptible variety under heavily infections. Okay, so this is the some historical concept or economic importance of this disease. Okay, so you see this healthy one okra plot. So this is the healthy one okra plants. Okay, so first of all, we see the symptoms of this virus seen on the okra crop. Okay, so you see these affected bhendi plants or okra plants. So initially, the affected leaf of this plant having leaf generally somewhat yellow and somewhat green colors is developed. Means the leaf lamina of this leaf generally green in color, and when of this affected leaf is yellow in colors. Okay, but under severe infection or under favorable conditions, this affected leaf become completely chlorotic in nature. Means day by day, these virus utilize the cell sap from this leaf. So this leaf lamina having chlorophyll content is reduced. So that's why this green color replaced and to form the yellow color. Means chlorotic leaf generally produced from the this infected bhindi plants. Okay, means the virus produces the typical when yellowing and thickening of the leaf. So these naturally two symptoms generally seen on this infected leaf. Okay, so this virus also infect to the petiole, stem portion, and also the fruits. Okay, suppose this virus get multiplied to hold this growing bhendi crops. So majorly symptoms seen on this plant, that is stunted the growth of these virus affected bhendi crops due to the shortening of this node portion. So that's why small plants, virus affected plants, is seen in the bhendi fields. Okay, so these affected plants produces only few number of the fruits, and these fruits are generally small in size and somewhat variegated symptoms are seen. Means some chlorotic patches are seen on the outer side of these fruits. Okay, so these infected plants produces only few number of the fruits. Okay, so this is the majorly symptoms seen on the bhendi plants due to the virus. Okay, so you also see this another pictorial or pictures regarding the yellow vein mosaic symptoms seen on the bhendi crops. Okay, so you see these first pictures in which near about 80 to 90% area of this leaf get infected by the virus and showing the yellow in colors. Then you see the second pictures. So this is the just the initial infection seen on this leaf of the bhendi. Means only few portion of the leaf or leaf lamina get infected and showing the chlorotic patches. Okay, so see these third pictures. So in these pictures, the leaf lamina generally green in color and remaining vein and veinlet of this leaf become yellow in colors. Okay, then you see these pictures. So maximum chlorotic patches seen on the lower side of this bhendi leaf. Okay, means variegated type of the symptoms majorly seen on the bhendi due to the virus infections. Okay, so this is the just infected virus plant seen in these bhendi fields. Okay, so this is the infected plants. Means these plants is very means just height of this plant is generally short because this virus generally infect to this younger seedlings of this bhendi crop. So that's why the height of the plant is also stunted. Okay. Then causal organisms. Okay. So this yellow vein mosaic virus generally infect with the bhendi crops. So name of this virus, that is a DNA virus. Okay, double stranded DNA virus. So you know about these etiological parts of this DNA virus. So this DNA virus seen in bhendi crops. So DNA is a monopartite satellite virus having single stranded DNA nucleic acid. Okay. So this DNA virus having the groups, that is a Bogomo virus, including the family Gemini viridae. Okay, means this DNA virus comes under the Gemini viridae family. Okay, so this virus mainly transmitted from one plant to the another plant or one field to the another field through insect vector. Okay, vector means any virus causing disease from one plant to the another plant. Okay, so this is known as the vector. So in this case, the white flies, scientific name of this white flies, that is Bemisia tabaci. So this Bemisia tabaci insect can transmit the DNA virus from infected bhendi crop to the healthy bhendi crops or plants. Okay, so this virus firstly multiply or survival on various weeds generally seen along the roadside or in the bunds of these bhendi fields, that is some croton species or agritum weed species seen near to the roadside or in the bhendi fields. Okay, means this virus survival on these weed species in the absence of the bhendi crops. Okay, this virus is also survival on the some collateral host, that is other Malvaceae family crops, that is hibiscus abelmoicheus. So hibiscus abelmoicheus, this is the ornamental plants. So this virus also survival on these ornamental plants, that is a collateral host, means hibiscus species. Okay, means in short, this virus is neither sap transmissible or nor seed transmissible. Means this virus cannot transmitted through sap or contact method or using the seeds. Means this virus just transmit from one plant to the another plant through insect vector, that is white flies. Okay. So in the absence of the bhendi crop, this virus multiply on the other malvaceous hamili crop, that is the hibiscus species, and some weed species. Okay. Then we go to the last point, that is the control measures. Means how to control this yellow vein mosaic disease in bhendi crops. Okay. So most effective management practices done by these farmers, that is destruction of weed host and other malvaceous host whenever possible should be given in importance, because this virus generally survival on some weed species and some other malvaceous host crop, that is hibiscus species, in the absence of the bhendi crops. So it is necessary to eradicate this weed host and malvaceous host near to the bhendi fields. Okay, then locate the virus infected bhendi plants and destroy properly, or rogue out this plant during the early growth of these bhendi crops. Okay, then the pumpkin crops is not grown near or as a mixed cropping in bhendi crops, because pumpkin is also host crop for survival of the virus. So avoid the growing of this pumpkin in between the bhendi rows. Okay, then planting of the 4 rows of tall border crops, that is maize, sorghum, or bajra crops around the field of the bhendi crops. So reduce the whitefly infestations and also the disease. Means you can sow the seed of the maize, sorghum, or pearl millet around the border of particularly bhendi fields. So this whitefly generally utilize the cell sap or attack to these border crops. Means to minimize the attack of these white flies on the main bhendi crops by growing the jawar, maize, or pearl millet around the field of these bhendi crops. Okay, then suppose the whitefly population is increased in particularly bhendi fields. So immediately spraying this bhendi crop with dimethoate 0.05% or endosulfan 0.06% or 0.05% methyl damatol or phosphimidone. So this systemic insecticide generally applied over this bhendi crop for reduce the population of the white flies. So ultimately you can reduce the yellow vein mosaic disease in bhendi crops. Okay. Then use of the yellow color polythene mulch deter away from the white flies, or use of the yellow sticky traps in open field as well as greenhouses, bendy crops. Okay, so this is the also most important management practices generally done by the farmers in greenhouse as well as open field. Okay, then release of Chrysoperla cornea at the rate of 800 larvae per acre. Chrysoperla cornea, this is the scientific name of green lacewing. Okay, means release of parasitide at the rate of 800 larvae per acre. So this is the beneficial to manage this yellow vein mosaic disease in bhendi crop. So this is the biological management point instead of this insecticides or chemical managements. Okay, and last point, growing resistant varieties like Arca anamica, Arca abhay, Varsha upahar, and some tolerant varieties, that is a Parvani Kranti. So this Parvani Kranti variety developed by the VNMKV Parvani. Okay, so in this way you can control or manage this yellow vein mosaic disease in bhendi crop. Okay, so this yellow vein mosaic disease is the most common and most destructive or devastating disease seen in bhendi crop. So you can properly manage by adopting some cultural management, some biological management, and some chemical management or growing resistant varieties. So you can properly control this virus seen in bhendi crops. Okay, so this is the brief information about the yellow vein mosaic disease seen in bhendi crops. Okay, thank you.